Hey, welcome back, biophysicists. Today we are with Jacob Cook, a student out of London that the recent paper in Plouse Computational Biology is on the thermodynamic constraints on the assembly and diversity of microbial ecosystems are different near to and far from equilibrium. Jacob, thanks for being on the show.
Well, thanks for having me on the show. It's a pleasure.
Great. Why don't we just go ahead and dive right on in then? So what kind of got you interested in studying microbial ecosystems?
So I sort of come at it more from thermodynamics than anything else. I sort of was quite interested towards the beginning of my PhD in kind of stochastic thermodynamics. And I got funding to sort of apply stochastic thermodynamics to ecology. And we originally did it from a very quite a theoretical perspective. So looked at really simple biochemical systems and looked at whether you could sort of derive sort of novel thermodynamic laws that could then sort of scale up to full scale ecosystems. And it wasn't a complete failure, but it was clearly never going to sort of scale to the sort of ecosystem level that I personally find quite interesting, thinking about like this sort of like quite complicated systems and how you describe them, how you
Yeah, I
model
mean,
them.
stochastic thermodynamics is nice in the sense that, you get kind of this average over all of these very, very noisy pieces. And I would imagine for these ecosystems, there's a lot of noisy pieces. So you have to have some framework to study this that's not just like a very simple classical like thing.
Yeah, exactly. Yeah, it's basically precisely that. There's just so many. Once you get to the microbial level, there's so many, just so many moving. You said noisy pieces, but yeah, exactly. Just so many of these pieces, and they intersect. And how do you even think of them in a sort of conceivable, in a sort of tractable manner? That's what I mean.
Okay, so
Um,
I guess like, you have this framework of stochastic thermodynamics that you want to apply to, and you have, your system of microbes. How do you even kind of start to go about creating a framework to study this?
so actually, the part of it is almost chucking out the stochastic thermodynamics. Is it just being like, I think it's just the realization you had halfway through the PhD where it's just like, if you could get it to work, it would be amazing. But it just doesn't feel like, it feels like something, if I was like, I'm going to dedicate the next 40 years of my life to this, it'd be a good aim rather than like, this is something I can do within the course of a sort of
Yeah, I
normal
mean,
PhD schedule.
when you're working on a degree, you don't want it to hear it's going to take 40 years. I don't think any
Yeah.
student walks out of their advisor's office and goes, "Yes, I'll be 65 when I'm done with this." So
Yeah.
why don't you describe then a little bit of the model and how you even start going about with this?
Yeah. So what I think what to give maybe give a bit of context of what I did instead, that might be sensible.
Yeah.
And I'd say the broad context is in there's data in microbial ecology is improved a lot, presumably as you would if you talk to other people who work on sort of physics and microbial ecology, you'll hear that a lot, that the data has become much better. And there's a sort of increase in the number of like novel modeling approaches coming along with that data. And I think the big thing for me, the paper I read was it's called like Goldford et al., which is a 2018 science paper. And in that paper, they basically show that you can match kind of using a sort of consumer resource model. You can which is basically a model where microbes both consume resources but also produce them. If you use these kind of consumer resource model with metabolite production, you can actually describe and match kind of empirical patterns of microbial communities that have been transplanted onto substrates. And I think that's that was quite a big result. I didn't clock it when I first read it. It was a big result because you just come out of like doing a physics degree and you're like, oh, you get model predictions right all of the time. Whereas actually taking a model that simple and being able to describe an empirical pattern that's like reproducible is actually quite a big thing. So I think that's sort of that's kind of how I came to it, is looking at these consumer resource models and being like, oh, these have had this like notable recent success. Should we think about incorporating like new elements in them? Are there things that could sort of additionally be put in that make them maybe more physically realistic, or maybe more better description? And that's kind of how I came to sort of came to thinking about the model, or if that makes sense.
Yeah, yeah. I mean, I guess we can even take a step back. I was racing ahead straight to the model, but can you give us like some type of framework for what this system even looks like? Like what exactly is a microbial ecosystem and what's interesting to study with it?
Yeah, so I think there's almost two schools of thought in terms of like modeling microbial ecosystems, and they're not schools of thought because they're not mutually exclusive, but there's two sort of broad streams in that there's thinking about all of the kind of cool and wacky stuff bacteria do, like produce phages. They're like machinery they use to inject other bacteria to kill them, and thinking about all of the complicated biophysics of this. And then there's the sort of like almost a purely metabolic line of thought where you just think about what is the bacteria eating, what does it then produce as a byproduct of having eaten that, and then how does that kind of processing of energy and nutrients, how does that sort of how well can you describe the system from that alone without thinking about wacky things like chemotaxis or type 6 injection machinery, all of those kind of very cool things they do, but how necessary are they to model a system? Like, do you need to understand them to understand what's going on in the human gut? Like, maybe you do at the edge of a wound where it's sort of a heterogeneous environment, you want to know which strain comes out on top, but to describe how the sort of community processes the substrate in the gut, you maybe don't need those things. So it's sort of that second, let's ignore all of the kind of potentially quite exciting details about bacteria and just focus on them purely as like these little metabolic machines. And that kind of lends itself to, it's not the most exciting biological thing, but from a thermodynamics perspective, you've got the kind of, these are little balls which thermodynamics is happening within. And that's kind of I don't know, I found that quite nice.
So it's kind of like I guess it's kind of like, the ensemble is I have like a little, I think Gibbs defined as like you can have an ensemble of ensembles, I guess. So you have like a little machine and then I just have a huge ensemble of these machines. And
yeah,
these
basically.
machines are kind of isolated in some sense. So I don't have any aliens, I guess, kind of coming in.
Yeah, and there's a sort of external chemical pressure. So this machine has the ability to turn from metabolite 1 to metabolite 3. And there's a kind of external pressure that the whole ensemble of these little machines are maintaining. And as the concentration of 1 and 3 change relative, that changes the rate at which they're processing the substrate and the rate at which they can obtain energy and therefore grow, sort of. And it's quite a reductionist view. We
Yeah,
assume
I
they
mean,
were.
of course, like, the human gut is extremely complex, so it's not like you can just kind of line up and take a quick peek at it. But it is interesting, I think, kind of these two ways that you described the modeling approach. I mean, this might be kind of a reach, but it sounds to me very similar to like kind of how they define what is life in some sense, like the characteristics that they look for it. So I think one is that they look for, as you said, kind of this metabolic pathway. And then the other one is like, can it reproduce itself? So, like, there's like the question of, is a virus really a living thing? It can reproduce, but it can't like reproduce itself. It has to have a host in order to do it. That's just a tangential thought that I had, more or less.
It's quite an interesting tangential thought. Like, yeah. Nice.
So I
Yeah.
guess, we have these little machines now that are running around doing their business. How do we go about describing how they do what they do?
Yeah. So that's the next part. So the sort of, we wanted to sort of think of a little model for this, and they kind of To think about them in terms of thermodynamics, the pre-existing kind of consumer resource model is quite natural in that you have a you have a you have a substrate that's consumed, and if you've got production of metabolites, that can then be thought of as being consumed into something else, and that sort of fits nicely with not necessarily stochastic thermodynamics because you're sort of taking a mean field limit, but sort of non-equilibrium thermodynamics where you think there's still going to, you are modeling both the forward rate of the process and the sort of backward rate. Sort of, well, basic chemical thermodynamics where everything is out of equilibrium. So you've got so that's the sort of natural, already a natural framework that you've got a substrate, and then the bacteria, if it has the right machinery to break down that substrate, it then breaks it down to a secondary metabolite. And then we sort of want to think about,
you have a full chain of these reactions that basically break down or build up certain materials.
yeah, so in our case, because this is again highly reductionist, you could in theory have the different chains of reactions that break down and build up, but we literally just think about the breaking down. So we sort of fudge it and remove some of the complexity by saying we're thinking about the breaking down pathway, which is like the obtaining of energy, and then this sort of energy currency is fed into a catabolism sort of building up pathway that we're gonna sort of fudge for now, because just as a sort of another reductionist step.
So it's basically like I can take the stairs down and I have very discrete points all the way, but when I get to the bottom, I can only take like an elevator. So
sort of. I think the idea is that the first metabolite is only supplied, nothing produces it. And then there's sort of elevators going off at right angles that produce various proteins that are needed to sustain the cell is kind of the idea. Although it would be interesting if they produced their own.
Yeah, I mean, because I would imagine you would have like all these different feedback loops, because in most enzymatic pathways, you know, they're massively complex. So I guess like how do you pick what's kind of important and interesting in these systems to take a look at?
Yeah, that is a good point. That's one of the things where this is something we got when we got reviewer comments. They were talking about quite a bit, was the idea of like integrating catabolism as well as no, am I getting this one way around? We have catabolism.
Anabolic is building, yeah.
Building, incorporating anabolism as well as catabolism, and sort of having the sort of interplay and the link and the feedback. And I think that's definitely something that if I was to build this model further, I'd really think about, because I think it is like the feedback would be very rich. But yeah, just for the sort of, we need to get a model working, I was just sort of at some point just cut off and never thought about the thermodynamics on that side.
Yeah, I mean, you always start with like the simplest possible like harmonic spring you can think of, and you can always add complexity on top of it. Yeah.
Yeah. So that was kind of,
I guess though, you know, when you strip this thing down as much as you possibly can, like, do you pick what to strip down based on like the complexity of modeling, or is there some like biological intuition you have that says that, okay, this process is like either the slow one or the more interesting one that we care to look at?
I think to some extent it was my level of satisfaction with pre-existing models, in that it's a really, really nice model of the proteome fraction dynamics that's sort of developed by various people, mainly Terry Hua, who's like a physicist who kind of went into sort of experimental cell biology, where you can kind of model, and this is part of the paper where we sort of use their model for proteome fraction dynamics, where you can shift between the two. And I just thought, oh, that's a really lovely modeling framework. And that sort of handled anabolism for us. And I think that's better. It wasn't really a kind of really considered decision not to include animalism. We just kind of handled it through this like really nice model that I really liked that we've come across, and sort of merged them together to have the sort of internal dynamics of anabolism coupled to the sort of thermodynamic oh god,
Catabolic,
catabolism
yeah.
model. Catabolic, that's it. And I think it just kind of grew like that more than any sort of solid, which I think is often the way. And then you look back and you're like, I just did this because of my sort of a priori biases towards, I thought this model was nice, rather than some kind of, yeah.
Yeah, I mean, it's kind of like a what hindsight bias. You're like, yeah, the path I took is the best, of course. Okay, but anyway, so now we have kind of some idea about what we're trying to model. You know, if you kind of put the pencil to the paper now, what are you trying to write down to describe? And I guess what are the factors that go into describing these different reactions?
So
I guess kind of
yeah,
maybe a better way to put that is, you know, what's the parameter set and parameter space that you're looking over?
yeah, I guess like, so there's two fundamental things that you'd want as a readout from a microbial ecosystem, which are the uh, the kind of growth rate of any given population and the consumption rate of any given substrate. Those are kind of, you might only be interested in one of them for a specific purpose, but in general, you're interested in the populations because they suggest what strains will come to dominate. And if you're interested in biodiversity, that matters. If you're interested in disease, that matters because you care about, does this particular strain come to dominate? And then for the more biotechy applications, you care about the sort of consumption or production rate of certain metabolites. So those are kind of the two things you're looking to looking as kind of readouts. And then I think it's thinking about what fundamentally constrains the growth rates. So what governs the strain growth rate? And with the sort of, if I start to go into kind of little protein dynamics model that we sort of attach in for this side, we sort of think of the key restrictions on the growth rate as being the rate at which energy is sort of available, because that can, if you've got a greater supply of energy, you can grow faster. If you're short of energy, you're not going to grow.
Mm-hmm.
And then the other thing from the protein dynamics model is that your rate of growth is constrained by the rate at which the sort of maximum number of ribosomes you have. Because if you've got there's only so fast that ribosomes can go. If you've got enough energy, they still can't translate like a million bases per second per ribosome. Like there's a hard limit there as well. And so that's kind of how we get to, in short, how we get to the sort of proteome dynamics thing, in that there's the energy constraint acts through the amount of protein you're allocating to enzymes to obtain energy, and then the sort of ribosome thing is basically how much you're allocating to ribosomes.
So yeah, I mean, I guess it's kind of like, if in some sense, I kind of like to think of it as like building a house, I guess, you know, like I have some money that I can spend, which in this case is the energy more or less, and how much money I have dictates how nice of a house, and then also how many builders are in the area dictates how fast I can actually put the house up, right? Because I can literally have infinite money, but if there's no one around to build the house is not getting built. So the ribosome is more or less the builder in this case.
Yeah, and then there's a bit of a trade-off, because if you had, I don't know, imagine if you're building a house but you're also running a business. So you need the money, you need the business to run. I suppose if you're running a building firm and you can change your builders from working on other people's houses to get you money, this is probably becoming a confused metaphor. But also the builders, you can either allocate the builders to help build your house as ribosomes, or you can send them out to different work sites and they'll get money for your business, if that makes sense.
Yeah,
So there's
yeah,
a sort of
you are the owner of the construction company. Yeah, I like
yeah,
this. It's a good metaphor.
and that's kind of yeah, and that's the sort of proteome trade-off is um,
So I
yeah.
guess, in the sense of the cell, what really, is the money, so to speak, is the ATP. So, right? So how do you go about treating the ATP of the cell?
Treating the ATP. Yeah. So I suppose in our model we would say there were two separate currencies, which is a slight there's the protein, so you are like every cell has a set not a set number, but a relatively fixed number of proteins. Or no, that's not correct. Any protein that's allocated to one task isn't allocated to another task. So there's a sort of opportunity cost there, even if you can have more. And then the other currency and the sort of the one we treat as sort of universal currency is ATP. And so the idea is that it's kind of coupled to ATP production is coupled to this sort of breakdown of metabolites. So you break down metabolites and that can liberate free energy, which you then use as ATP. And then using that ATP, yeah, what am I going to say? I guess.
So I have something comes in, I have like the big piece that I'm gonna break down into the next chunk or the next little bit of it. So when that happens, I release energy, right?
Mhm.
And that energy can go into creating ATP.
Yeah, so I think the idea is that in a natural cell, what you'd be doing is you'd have your kind of your metabolic process, your enzyme reaction, and that there'll be an intrinsic coupling to some ATP production. And then the cell could also tune its production of ATP a bit by using extra liberated free energy to pump ions across a membrane. And then that sort of returns, you can return that to the sort of universal currency by undoing pumping over the enzyme and using that to generate ATP. So there's a bit of kind of, there can be a little bit of tuning to ensure that you're producing sort of more thermodynamically optimal kind of breakdown of metabolites.
And that ATP then gets used with the ribosome to create your proteins.
Yeah, exactly. And so we don't have anything particularly dynamic on that side. It's just that you need this buildup of ATP to keep the ribosomes translating is the sort of dynamic there.
Okay, so if you completely run out of ATP, your ribosome more or less just stalls.
Yeah, and there's a kind of dependence on, it's a bit unclear because these things are very hard to probe, but there's thought to be some level of dependence on ribosome translation rate and the availability of cellular energy. It isn't kind of core to the results of our paper, but there is thought to be, if you drop below a certain ATP level, it starts to go slower and slower and then it stalls.
I mean, I guess going back to the construction analogy, it's like reducing the wages of your workers lower and lower. At some point, they're going to strike and then stop working altogether.
Yeah, that does work.
But I guess, so I found one kind of interesting thing in the paper is you kind of derive this reversible kinetic schema that takes into account this fact that, my reactions can kind of leap forward and go backwards. So you make kind of this nice parallel between this what's called the thermodynamic factor and the Michaelis-Menten kinetics. Can you talk a little bit about how this is kind of an extension or I guess, yeah, more or less like kind of an extension of the Michaelis-Menten idea?
Yeah, so it's something I've more adopted. It wasn't really something I derived, or the derivation is very old, and it's kind of a derivation I've taken and kind of adapted to this circumstance more than something I derived originally. But because it comes from sort of like Haldane, so that kind of era of sort of early molecular biology, basically, or maybe before they even knew there were molecules, but that kind of very early
Mhm.
era. Um, and the idea is basically that one of the assumptions that the Michaelis-Menten kinetics make are that your reaction is sufficiently far from equilibrium that there are no thermodynamic factors. And this is quite often a good assumption. Like this is, if you're growing strains in a lab on glucose, the amount of free energy in kind of glucose reactions and is just so great that you're not you will have a reverse rate, but like if it reverse rate is happening at 10 to the minus 20 times of the forward rate, like who cares? And you kind of is this is sort of something that's interesting with protein synthesis is that the energy contained in ATP is so high, there's so much free energy there that when you're burning one ATP, you're just quite often you're just chucking so much energy at the problem that you don't really have to worry about the reverse rate. So that's kind of an interesting, like in terms of the sort of protein synthesis, using a sort of Michaelis-Menten reaction kind of is actually kind of reasonable. Like it's, you're just using so much free energy that you don't have to worry about the reverse rate in a way you would with sort of stochastic thermodynamics, or stochastic thermodynamics is entirely about the reverse rate being very significant. And I think the idea is that when you use this kind of form with the thermodynamic factor, you can capture situations where you are actually using reactions that are much closer to equilibrium. So it's been applied a little bit more in ecology when they do kind of modeling of methanogenic reactors. So you have a load of sewage and you want to keep a production of methane that can then be burnt off as a kind of greenish kind of, well, it's greener than letting the methane escape, basically. And a sort of greenish solution to energy, you have these reactors where you've got microbial communities that are producing methane from kind of slurry, sewage, things of that nature. And the idea there is that those are actually communities where they are pretty close to thermodynamic equilibrium in the reactions that they're subsisting off. And so that's kind of why thinking about using this sort of scheme that includes a thermodynamic factor, which basically accounts for as you get closer to thermodynamic equilibrium, you're maintaining a good forward rate, but it's beginning to be cancelled out by the backwards rate. And when you hit equilibrium, you've still got this pretty good forward rate, but you've also got an equal backwards rate. So net, you're not actually producing any methane or whatever they're doing. So the sort of about the reason to use it, or the argument I would make, is thinking about circumstances that are common in real ecosystems where you don't have this really great high free energy source that just can really easily grow off. And it's actually trying to describe those situations, not just a lab situation where you're basically picking a culture medium specifically for how good it is to grow off, which means you're not limited by, people usually say things like you're not limited by nitrogen or phosphorus, but also you're going to provide something that's got absolutely shitloads of free energy, just loads and loads of it. And I think thinking about what happens when you are thinking of situations where there probably is some degree of thermodynamic limitation is kind of a nice sort of the reason I do it,
So
or the
yeah, I
reason we did
mean,
do
I
it.
guess you don't just have like all this available energy for the cell to use. They have to be a little more clever with how they proceed in their reactions, because if I expend a lot of energy to make something, well, it can turn right around and kind of come back, and I end up more or less where I started from. Is that
Yeah,
fair to say?
exactly. Yeah, I think that's completely fair. And everything becomes much more context dependent, because if there's a huge amount of free energy available, then it doesn't really matter how much waste is built up or things like that. Whereas if you're actually sort of just getting by in terms of the reactions proceeding just enough for you to actually grow on, only small changes to environmental concentrations of other metabolites can just screw up your pathway. And that's quite important for a lot of the real world pathways are actually quite sensitive to small changes in oxygen, pH, things like nitrification in soil, things like this. A lot of the things that microbes do that are quite important are actually very, very sensitive to small changes, which is kind of, I'm not sure if it's necessarily thermodynamics. I wouldn't quite go as far as saying that, but it is sort of indicative that maybe there is something thermodynamic, some kind of limitation here going on. The fact that pH doesn't have to change that much for suddenly to be producing way more nitrous oxide from the soil. Like it's
Yeah, I mean, biology is usually pretty good at fine tuning something to a particular like stable point. So it's not super surprising to me to hear that, you know, if I push something, perturb it a little bit, I can wind up in a completely new like stable state.
yeah,
So
that's
I guess
yeah.
kind of like what's kind of the main takeaway now that if I incorporate this reversible kinetics into my reaction, what do I see now that I probably wouldn't have seen before?
So if you look at a kind of high free energy case, you wouldn't see anything new. You just see it's just the same as ever, really. Like, and that's one of the conclusions. It's like you include it at high free energy, and then it's actually just, you might as well use Nicholas Menten. It turns out they were onto something. And then I think the thing is, it's thinking about the low free energy case. And I think one of the things we find is that sort of different from what you'd expect with a model not including this is one thing I found that's cool anyway is that you end up with, you can see that microbial species don't necessarily just interact in the conventional ways people think about, which is competing for a substrate. So you can have an impact on a different microbial species because you're both competing for the same substrate. That's sort of known to happen. And then you've also got this kind of cross-feeding where you are microbial species 1, you produce a waste product, microbial species 2 eats this product. And so you see this kind of positive, if you've got microbial species 1, then it has a positive effect on microbial species 2. And I think it was quite nice that you can show that you also get these other kinds of competition where 2 things are pumping out the same waste product, and you can get an adverse, so the 2 species consume completely different substrates, but they're still competing through their metabolism, even though they're not consuming the same substrates, because they're both producing waste products that inhibit the consumption of their particular substrates.
So it's like if, you know, you and your roommate or you and your flatmate, you both eat different stuff, but you use the same trash can, how quickly that fills up. Yeah.
Yeah, absolutely. Yeah. And I think that's quite a nice result that you can, well, not result, but it's just interesting in terms of you can see that even just considering metabolism, you can interact in more ways than just substrate competition. It's a weaker effect, but they can, in some cases, it can be quite strong. It can be of the same order of sort of substrate competition and stuff.
Yeah, I mean, it's definitely putting like kind of I guess, a reverse-ish pressure on the system, because you would think it's only what I take in, but it's also kind of what you put out as well.
Yeah. And also the other one is that you can take someone's waste and break it down. And that doesn't just help you because you're like, I've got something to eat. That helps them because you're breaking down their products as well. And that's the other one that's kind of, I think that's quite.
So you and your flatmate got a dog then, and he just gets in the trash all the time.
Exactly.
So I guess one thing that I kind of found interesting about this is you found kind of this nice metric that you can use to kind of quantify how these reactions progress through kind of an entropy production rate. But
Mhm.
what was interesting about this, and maybe you can just speak some more about the entropy production rate in general, is how you can relate that to kind of the classical thermodynamic of entropy production is like change in heat over the temperature.
Yeah. So this is on, what is it, page
This is page 8, I think.
8? Yeah. So
Equation 14.
so it's kind of, yeah, exactly how it relates to heat. I'm not 100%. I feel like heat is the most likely form that this entropy would result in, and this would be more significant than the sort of changing chemical form of the material.
I mean, entropy can take a bunch of different forms. It's just the easiest one I think it always reverts back to is heat.
To heat, yeah. So because
I'm
we
just
sort of
kind of
claim that
curious about this entropy production rate in general, like, kind of how you came about getting this and how you can use it to kind of track your system in general.
yeah, so it kind of just looks like doing, it was more just from kind of Gibbs free energy accounting than anything else. So it's just sort of thinking about how the initial substrate has a kind of associated Gibbs free energy to do, and then the final product has a sort of its own associated Gibbs free energy, and then you have an obvious destination for any Gibbs free energy that you want to transduce, which is the ATP, and you end up with, if you think about the Gibbs free energy would be, I don't remember the exact, what have I got to here? And then you just end up with a kind of Gibbs free energy change of whatever hasn't gone. You've got a certain reaction rate, which is known from your sort of reaction kinetics. Whatever Gibbs free energy has not been stored as ATP has to have been dissipated, because you know there's a change in the Gibbs. And
So it's just essentially like a free energy balance. So you take like
yeah,
the
just
log ratio of your products and reactants, you take how much free energy comes from how much free energy is moving around with the ATP, and the bit that's left over, well, that has to have gone somewhere.
exactly, yeah.
So I
And
guess, now that we look at kind of this change in free energy and we can relate that to an entropy production rate, what does this entropy production rate tell us in terms of the system?
so I guess there's a few things it tells us. We make this claim that you can maybe measure the heat production, and that could tell you something about how fast the system's developing, because you see clear spikes kind of as you've got a substrate. We have a substrate that's kind of the highest level substrate, and it kind of sequentially differentiates into more and more substrates. And when you get the sort of rapid periods of growth, they're associated with like suddenly a new substrate becoming available. And so the idea is that you could almost track that if you were looking at a situation like this, you could almost track the development of the system through bursts of heat production. Whether or not that's actually empirically feasible it was kind of something we just threw in there because it sounded
I mean,
cool.
it sounds very similar to isothermal titration calorimetry. So there, I'm not sure if you're familiar with it, but I could be misquoting this, but I think the general idea is I have a solution that I closely am monitoring the temperature, and I take another molecule and I throw it in there. And as this thing reacts, it gives off or takes away certain amounts of heat. And you can track that amount of heat as the reaction progresses. And it tells you more or less like how the entropy and the free energy are changing during the course of that reaction.
I had not come across that, but that does sound like the kind of thing we'd be arguing that you could do. So I think
So, I
kind of
mean, yeah,
interesting.
like it sounds feasible, like, but like you said, whether or not this free energy is turned into heat is I guess, a different kind of debate.
Yeah. And then there's a kind of another aspect to that where there's been a kind of recent hypothesis. I can't remember the paper. I think they were like Niebel et al. It's one of the references where they hypothesize this sort of that there's an upper rate in terms of how much entropy the cells can remove. So when you have entropy produced in the cell, this is obviously a problem for the cell because it's heat, it's disordered molecules. And the idea that there's an upper rate which actually acts as a constraint on the sort of maximum rate that a cell can process a substrate is constrained not just by like sort of biophysical constraints. It's also the rate at which they can remove the entropy that's produced, which is probably a biophysical constraint. So I probably should ignore that thing I said.
Yeah.
But as a
I
kind of
mean, yeah,
additional constraint.
because you can only dissipate heat so fast, right? It's not like you can just open the window of a cell and let it kind of flush out.
Yeah, and it's interesting. It seems to have it's been observed in a couple of, or at least one place, and it's kind of an interesting. Yeah, it's one of those things where it's hard to work out whether it would be the case because there is a limit to how fast you dissipate heat, but whether it's relevant for like these tiny cells is an interesting one.
Yeah,
I
I
guess.
mean, that's almost more of like a, I don't want to say an engineering problem, but if you have a sphere that's a certain temperature, like how quickly does the heat radiate out from it? You need to know all the different, like, what's the convective flow over the cell? Like, there's a whole nother host of problems that come with that.
And like colony morphology, so how close are the cells to each other? Because if you've got a whole bunch of them that are doing this, it's going to be very different.
This sounds like the topic of someone else's thesis. So I guess what are kind of the main assumptions behind the model that you make then?
There are, oh God, I've got a whole, I end up writing, there's quite a lot of assumptions. One of the reviewers asked us to write out all the assumptions of the model, and it ended up as a supplementary table, which is 1, 2, 3, something like 3 pages long.
Okay.
I think there's a few.
I
There's
guess
a lot.
what are the biggest key assumptions then?
What's the biggest key assumption? The one we talked about previously is that you only have to think about the thermodynamic behavior of catabolism and not anabolism. I think that's a big assumption, and that's something I would, if I was to have my time again on this project, I would try and rectify. I think that's one of the biggest ones. What are the other ones? I'm literally just scrolling through. I think one of the biggest assumptions we make is about the almost the structure of the substrate you supplied. Because all our results are you supply a substrate and it can be broken down, but it has to be broken down in these little steps, if that makes sense. I think this is somewhere in figure 1, you've got that kind of diagonal and you have to descend the diagonal in order to kind of, which basically bans any species from having a reaction that takes you from the best substrate to a waste product. They can have it, but it requires them to express like 6 reactions that take you each step. So you go from 1 to 3, 3 to 5, 5 to 7, if that makes sense. So it's more, I think that's a big assumption. And we kind of, it's one of those things where we just started implementing it and it kind of made sense at the time. And then maybe it'd be very different in if we used a different structure of possible reactions. So I suspect our results are a bit dependent on that as a kind of.
So I guess kind of the reaction matrix, changing the shape of that maybe could change the constraints that you see.
Yeah, and the reason we didn't change it was because it's hard to work out how to do it in a reasonable manner. So we've got something that's kind of a bit unsatisfactory, but uh, the change to make it something that we're fully satisfied with um, is hard to do, because it's like as soon as you allow reactions that do more steps, the reactions that do more steps, they yield more free energy, so they're always going to be favored. And like, so you have to introduce something that disfavors them, but you can't really do that from first principles.
Yeah, I mean, it just sounds like you have to introduce a whole bunch of other parameters, I
Yeah,
guess, tune it in.
yeah. So I need to think about, yeah. It's one of the things that long term I'd like to, if I was to work further on this, change, but it's
If you're the 40-year PhD student, then there's time to think about it.
yeah, exactly.
So I guess, is there any other big surprising facts you found? Or things that you maybe weren't considering that kind of popped out from the results of the model?
um, what was I? Do I remember what surprised me here? I think. I think of something. Yeah, I think I can't think of anything that immediately surprised me, but it's hard to remember what surprised you, isn't it?
Yeah, I mean, it's all it all blurs together. Then I guess maybe another good way to say it then is, if you were to come up to like a lay person on the street, as you know, what would kind of be your
What would
big,
be my big?
sentence that, you put it on a billboard, you're like, this is what we found.
I guess I would say that um, that I think the thing we found is that it is it is plausible and it is sensible to think about as microbials as actually interacting and operating on like a thermodynamic level. And I think the interaction stuff is more it's more from a kind of ecological perspective than a physics perspective. This is maybe interesting, but I suppose they link together nicely. But it's that actually, if you want to think about how they're interacting, you do have to consider thermodynamics. I think that's kind of what I'd probably sell it based on, if that makes sense. And that these are kind of
The
it
coupling
is
of ecology and thermodynamics is a lot stronger than initially thought.
yeah, I think that would be my kind of take home. And you can ignore it if you choose the right limit, but it is like worth being aware of and thinking of. And it's not it's neither strong enough to determine it completely. Like it's one factor among many for sure, but it's not weak enough that it can be safely ignored. I
I
think that would
guess
be
it
my
kind of like relativity in some sense, right? If you find, if you stay here on Earth, you're fine with Newtonian mechanics, but the minute you reach a different limit, you have to apply like a new set of physics to understand it.
yeah. I think that's a very nice way of putting it. Yeah,
I mean,
I
it's
think
always nice to have a parallel between your work and Einstein too. So.
yeah. So I'm going to reuse that to make myself.
Okay, so I guess my last question then is, say you were to completely restart on this project, what would you do different?
Right. So I've got almost two levels to answer the question. I've got like the level which is like, what are the more sort of technical details and what I would change about it as a program of research. I don't know which is better to state, or just both, maybe.
Maybe the second one, kind of
The
the
second one.
bigger idea.
So I think like the sort of one of the underlying ideas in this model is the idea of like sort of cellular economics. So it's the idea that cells have kind of a definite currency that they can sort of choose to spend. And this comes under fire quite a lot from biochemists who go, well, it doesn't really work because actually biological systems aren't very efficient. Like things like protein translation operates nowhere near thermodynamic kind of optimality. You're just burning absolutely loads of ATP in order to kind of make sure you don't get copy errors. And so that's kind of the standard charge levied against any of this. And I think what's interesting and what I'd want to incorporate or further extend is thinking about what is it worth spending money on? So actually trying to extend the economics of thinking about not just like you spend, because in the model at the moment, it's like you get this energy and there's all this thermodynamics, which eventually results in this energy currency, which you spend synthesizing protein. And like thinking about how can you extend it to also incorporate things like nitrogen acquisition? So you need specific resources to grow. And then there's a trade-off between how much of your free energy you're prepared to kind of, from a thermodynamic perspective, waste, but from a perspective of like optimal growth or something, you're kind of investing this free energy by you're being inefficient, but it's letting you uptake nitrogen and stuff. Because it's quite interesting that when you look, if you look for, there's some like phenomenally inefficient microbes, and they almost always, there's one called Prococulus, which is a very common phytoplankton. It's like 25% of the total photosynthesis goes on in a single phytoplankton species, genus, I think. And like what it lives in a really, really nutrient poor part of the ocean, and it just spits out carbon, just like literally spits out huge amounts of carbon. And it's so that it basically its metabolism is running so fast that it can just like any nitrogen it takes up, it can just kind of cling on to, because it's got a really, and I think it's interesting that there's a few other cases like that where you get really inefficient microbes, and usually it's trying to hang on to something like iron or something that's scarce. So I've probably gone a slightly too blue skies thinking here, but like the idea of what are you going to spend this free energy on,
So
and
it's
how
like setting up a different type of optimization then, in some sense.
Yeah, that thermodynamics kind of feeds into as one aspect, in that this is the fundamental constraint on the sort of dynamics of your currency. And then how do you link that currency to a sort of broader idea of optimization, like optimal growth or optimal fecundity or something like that?
Yeah, if you have multiple kind of competing optimization problems.
Yeah, and I think that's what could take an idea of, because obviously efficiency does matter. Like I think I don't need to convince you or anyone who's probably listened to this podcast that efficiency matters, because it's pretty fundamental to the development of modern physics, the idea of it, and all of engineering, isn't it?
Anyone
but
that's
like
ever ridden a bike with flat tires knows full well how important efficiency is.
Yeah, and I think it's just taking this sort of like, what is it that makes it kind of a hard concept to apply, at least at a higher level in biology? I don't think anyone would dispute that it matters at like certain sort of molecular levels, and like what stops it from being a good framework to think about the development of biology as a whole, rather than just these specific cases where, oh, this type of information transmission is almost perfectly efficient, which definitely happens. But what things do you have to include as sort of multiple optimizations to think about, well, like ecology or evolution in that kind of efficiency framework?
What's the balance between each of those optimizations too, I guess, in some sense?
Yeah, and I think that's probably the $1 million question. That's the thing that makes it an incredibly long-term research project rather than
Yeah, I mean, that's the next grant writing session, I guess.
Yeah, the one after and the one after.
Okay. So is there any social media or something like that you want people to follow you on, or
Um,
just have a look at the paper?
just have a look at the paper. I have a academic Twitter account, but I barely ever use it apart from to tweet out my papers and then retweet people I know's papers. But I think what am I on Twitter? Give me a second.
This is how you know if it's not used a lot, when you actually have to look it up before you send it out.
Oh yeah, it's literally just at Jacob Cook 1995. So if anyone wants to follow me there, I'll happy to.
Okey-doke.
Also, if anyone wants to email me or anything with questions, I quite like getting emails, and that's just I don't know if that should be posted publicly, but it's attached to the paper. So if anyone feels like emailing me, then
I'll
go
I'll
ahead.
put your Twitter handle in the description, but I'll leave it to the listener to go look up your email. So
Yeah, because I'm worried. Like, yeah, it seems like a I don't know, asking to get emailed by anti-vaxxers or something. Which
yeah, some South Nigerian prince, I think, is struck upon some gold, but um
I get. Yeah. Anyway, sorry.
uh, no, but yeah, thanks so much for being on the show, Jacob. It's been a lot of fun.
That's all right. Thanks a lot. I've enjoyed chatting about it.